Hey there, it's Dr. Guerrero. I just wanted to give you a quick review for your upcoming lab exam, covering the labs that we did for Module 2. So, there we go. For Lab 6, which was in the actual lab manual, make sure you're familiar with the scientists associated with the cell theory. Know the difference between plant and animal cells, between prokaryotic and eukaryotic cells, so what those differences are. What cells in lab were used as examples specifically? What plant cells do we use? What animal cell do we use? What prokaryotic cells? Which eukaryotic cells do we use in lab? You need to be able to recognize and label all the bacteria that you looked at underneath the microscope by name. So what it was labeled on that microscope, and then like by classification, was it a cyanobacteria? And that is going to require you to look back into your lab manual for that information, and as well as labeling all the models that were used in the lab. So I did put together a little bit of information here so that to kind of jog your memory in case maybe you didn't get all of it in lab. But the scientists that factored into the cell theory, there were two that we talked about that invented microscopes of two different types. What specifically were those scientists' names and what did they look at? Okay. Then as we get into the cell theory, one of the first parts of the cell theory is that all living things are made of cells. And that was actually concluded by two different scientists. One was a botanist and one was a zoologist. And we had talked about that in lecture. So kind of left you some blanks there to kind of guide you. The second component of the cell theory is that the cell is the basic structural and functional unit of life. Then the third and final component of the cell theory is that all cells come from pre-existing cells. And again, I want you to know what scientist said that. Okay. Difference between plant and animal cells. These two diagrams are used just here in this review, but you and I both know, going through lecture, looking online, even looking within the lab manual, there are many different images that may be used. Be familiar with them. What makes a plant cell a plant cell? What makes an animal cell an animal cell? So what are some characteristics? Something that we would find only unique in animal cells, and something we would find only unique in plant cells. Okay, so helping us distinguish that these two types of cells are what they are based on these characteristics. Also, you should know that both plant and animal cells are eukaryotic cells. All right, so then we move on to the difference between a prokaryotic cell and a eukaryotic cell. I pulled these slides straight from your lecture, so you had some information that should look familiar to you, some characteristics that we discussed openly and specifically in our lecture when comparing both prokaryotic and eukaryotic cells. Then I went ahead and took some pictures of the microscopic images, but I didn't label them because these are things that I asked you to do in lab. And your images may look more clear, or you may have had a closer view, especially of this one here in the middle, and that is okay. I need to know, you should know that all of these are prokaryotes. Okay, these are all prokaryotes, and the only thing on earth that's prokaryotic is, I'm gonna let you fill in that blank. Then you should be able to label these based on the label that was put under the microscope so that you knew what you were looking at. You need to know the shapes of the bacteria. So what shape are they? You can get this from your lab manual as well. And if, like, for example, we were looking at an amoeba, which is the name of this bacteria, specifically what type of bacteria is it? Again, you can pull that information from the lab manual. I went ahead also and took pictures of the models as well as the diagrams that go along with them, to kind of put them in here. You should also have these. I asked you to get them, but in case you didn't, these will help with reference points, making sure you can label these models because you'll see them again in the lab exam. Here are the others. I'm going to go back a slide real quick. Yes. So knowing that this one here on the left, this is a prokaryotic standard bacteria that's being used here. Right here, it's labeled as an amoeba. So this is an amoeba, and you have the different components of the amoeba labeled. Then we have the paramecium. Paramecium is a protist. Amoeba is also a protist. Here we have it labeled as well. And then right here we have a euglena. Euglena is also a protist. So remember that bacteria, the prokaryotic cells are only bacterial, but the amoeba, the paramecium, and the euglena are all protists, and protists are eukaryotic cells. In this slides for lab 6, what animal cell specifically did we look at in this lab? What indicator did we use to stain it? And what did you notice about this once you were able to focus it under the microscope? What are characteristics? What did it look like? What did you have to identify? I will tell you, we will have the picture of what you looked at up on the slide for the lab exam. So you need to be able to identify it as well. So making sure you know what animal cell it was, where it came from, what indicators we used, and what characteristics that kind of stood out. And all of those were questions within the lab. What plant cell did we look at in this lab? And actually we looked at 2 plant cells, okay? That in class we discussed the differences because one of them was green and one of them was not green. And you can look back into your lab manual and it talks about the differences and guides you through it. I'm just trying to jog your memory in preparing for this exam. Did we use an indicator or stain? And if so, which one? And what are some things or characteristics that stood out about those plant cells you looked at? All right, so then the second / third lab that you did was 7.3, 7.4. That's when we did the osmosis. So the movement of water. So explaining and demonstrating osmosis, know this lab really well. There's quite a few questions about it on your lab exam. Explain and demonstrate the difference between tonicities. So hypertonic, hypotonic, isotonic. I talked about this again heavy in lecture as well. Be able to list any stains or indicators that were used during this lab and know what probes were used during this lab. Okay, if there were any probes used, and explaining the associated charge. So 7.3, 7.4 were the ones that were right outside of the lab manual. And then we went into diffusion through membrane. So I've kind of broken up those objectives to explain it in this way. So knowing the difference between osmosis and diffusion by definition. All right, and I have 2 definitions here for you to just look over and make sure you can identify. Then I want you to be able to tell me in lab 7.3, what animal cell did we use? What did we do to prepare the animal cells before we put them on the microscope slides? Okay. And then be able to explain in these animal cells what hypertonic looks like, and maybe some words that we used with that, what hypotonic looks like, and what isotonic looks like. Again, discussed really well in lecture. Remember the terms hemolysis or hemolysis, and then even the word crenation, being able to bring all of that back for this content. Then in lab 7.4, we looked at a plant cell. So what plant cell did we look at in this lab? How did we prepare that cell before we put it on the slide? Explain what hyper, hypo, and isotonic look like in this plant cell. This is another concept, again, we discussed really heavy in lecture. Know what Turner pressure is. Okay, why is that important? And know what plasmolysis or plasmolysis is and what it looks like what causes it. Okay. Then the final aspect or final part of the labs that we did for this quick turnover for a lab exam was diffusion through membranes. This is the one that you wrote up in your composition book. Okay, so starting off explaining permeability. What is permeability? What does semi-permeable mean? What does non-permeable mean? Or impermeable may also be used. The dialysis tubing. So what is it, first of all? What is it permeable to? And what is it impermeable or not permeable to? Know what probe we used in this lab. Explain how we use that probe. And the data was put off in charts. And we talked about this in class. So know how to read that. Name the specific probe and what it measured. So the name of the actual probe and what did it measure. And then which setup. There was multiple setups in this lab. Which setup showed the largest M, that largest M value, which was the slope, and why? Why did it have the largest M value? Okay, which one didn't have a large M, and why would that be? Be able to compare that information, have conversation about it. Okay, that gives you the content for lab exam 2. I hope that you enjoy your day and this helps you in preparing to be successful on this exam. Have a great day.